Okay, so it's been 3 days since we talked about it. We were talking about cell communication and ways that cells signal each other. So take a minute, talk to the people around you, communicate with those around you, and see if you can answer these questions. An example of direct contact: what is a ligand, and what are the 3 stages of the cell signaling pathway? Okay, somebody give me an example of direct contact cell communication. Gap junctions, what kind of cells have gap junctions? Animal cells, what do plant cells use? Plasmodesmata. You're chatty today, nice. What is a ligand? Or a ligand, however you like to say it. It's a signaling molecule.
Changes the shape. It changes the shape of the receptor protein and activates it.
So when it binds to the receptor, it's gonna change the shape of the receptor to cause some kind of reaction, some kind of pathway, which we'll talk about. And it's usually much smaller than the receptor that it's binding to. So it's usually a small molecule binding to a big molecule. And what are the 3 stages of the cell signaling pathway? Number 1, reception, signal reception. Number 2, signal transduction. Number 3, cellular response. Good. Okay, so we went through some stuff on Thursday. Oops. Hello, DNA. We went through some things on Friday, and I looked back at the picture for the GPCR that we were talking about, and I didn't like the second picture. There was something weird going on, glitchy with the graphic. So I just want to go through this really quickly, because the second picture I think is clearer now. Okay, so this is the GPCR. It's inactive. This is the G protein. So this is a receptor. This is the G protein. It's inactive. How do we know it? G is the signaling molecule binds to this GPCR. You can see how it's changed shape because I used a different graphic from a different textbook or a different edition of the textbook. But now that activates the G protein, which happens because now it's bound to what? Instead of GDP being bound, the G protein is activated because GTP is bound. So the diagram that we had before had like multiple GTPs and GDPs going on. So that GDP is off, the GTP is on. Now we have an activated G protein. That activated G protein will then dissociate from the G PCR, and it's going to move down to bind with the enzyme, and that's going to activate the enzyme. So this is an activated enzyme. GT, when the protein bonds, it's going to cause some kind of cellular response. The signaling molecule can then dissociate because it's not a permanent chain, it's not a permanent bond. That can dissociate and go back and be used again. So this can keep going. And this then over here, the G protein can act as a GTPase. So it will, this bonding is temporary between the G protein and the enzyme. So when it dissociates, this G protein can act as GTPase, which is going to inactivate the G protein. So it's going to remove the GTP. Because it's GTPase, it's going to remove the GTP and bond a GDP to it. And that is going to make it inactive. And that also can be recycled to use again up here. So I just felt like that diagram was confusing because of all the weird stuff that was going on. So that's a quick review of that. So we're going to talk about a couple more kinds of reception. So again, we're still kind of in this first step. We're not really talking about what happens in terms of signal transduction. We're talking about what happens in terms of signal reception. So there are three main types of cell surface transmembrane proteins. GPCR is one of them. The second kind of transmembrane protein that is important for cellular reception is a receptor tyrosine kinase, or an RTK. We have lots of acronyms this chapter. Okay, again, we're going to go through these kind of quickly. We've got some similarities. We have a ligand molecule coming in. These are our receptor tyrosine kinase molecules. Right now, they are inactive. I don't know why I drew another arrow there. And they are monomers. So they are single units. They've got binding sites, both of them, on the extracellular side. And on the intracellular side, they have these tyrosines. So that's kind of our setup. We have these transmembrane proteins with binding sites on the extracellular side, and we've got tyrosine tails on the intracellular side, and they're not active. So the ligand molecule is going to come in, the signal molecule is going to come in, and it's going to bind. And when it binds, it's going to form a dimer. This is called dimerization. So the two monomers are going to come together. So when those signal molecules bind at the binding sites, it activates them and causes them to dimerize. So they're going to come together. So now we have these two units together. Okay, this is going to activate. So this is now activated. When this is activated, see, it's got a nice yellow glow around it. The tyrosine region is really what's activated, and it's going to phosphorylate. So it's going to take phosphate groups from ATP and making them ADP, and these are going to attach to the different tyrosine. Anyone know what tyrosine is? What kind of molecule is tyrosine? It's amino acid. So these are specific amino acids on these proteins that will phosphorylate. So they're going to have phosphate groups added to them. So now we have a phosphorylated dimer. And it's fully activated. Once it's fully activated, then these proteins are going to come in. These are relay proteins. So these are activated relay proteins. Oops. And each one of them, these are inactive, sorry, down here. So they're inactive, and then when they bind to these phosphorylated tyrosines, they become activated. And they're each going to cause a cellular response. And they might cause different cellular responses. So when these relay proteins bind, they change shape to activate, and that allows a cellular response to happen. So we're using the term kinase, which I want to define really quickly. Kinase tells us it's what? ACE tells us it's an enzyme. It's gonna act on proteins. So it's going to. Sorry, I don't know why it's doing that. Catalyze the transfer of phosphate groups from ATP to a protein. So it's a generic term. When we talk about a protein kinase, it's taking the phosphate group from an ATP and attaching it to a protein. A receptor tyrosine kinase is a specific kind of protein kinase. What do you think it does that's specific? Yeah, so a protein kinase is going to attach those phosphate groups to a protein. A tyrosine kinase is going to attach those phosphate groups to tyrosine specifically, so to a specific amino acid on that protein. So receptor tyrosine kinase tells us it's a receptor. And it's going to bind the phosphate groups to tyrosine specifically. When we talk about these different kinds of receptors, I don't think we talked about that. I'm going to just talk about that too. Sometimes they don't work right, and when they don't work right, that's a problem because messages aren't getting sent, which means cellular responses are not happening right. So in the GPCRs, some bacteria, so going back a slide in our heads, some bacteria in GPCRs, some bacteria produce toxins, and those toxins will actually block the GPCRs, like cholera, which hopefully you've never had, whooping cough, which you probably never had either, botulism, hopefully also you've never experienced botulism, but all of those bacteria will produce toxins that will block the GPCRs. They interfere with them so they don't work. And about 60% of medicines, which is a pretty high percentage, about 60% of medicines actually influence GPCRs in some way. Okay, they're significant. Receptor tyrosine kinases, if they do not work right, they can be associated with a lot of different things. One of those is HER2, which is associated with breast cancer. I don't know if I'm guessing everyone in here probably has known someone who's had breast cancer, but one of the things they test for is HER2, if it's HER2 positive or HER2 negative. And HER2 positive cancers, breast cancers, are a little bit harder to treat because they have an RTK that is not functioning properly. That cancer causes the RTK to not function properly, so it's harder to treat. So that's one of the factors that they'll test for when someone gets a biopsy for breast cancer, is looking for if it's HER2 positive or HER2 negative. Okay, the third kind of receptors, transmembrane receptors that we're going to talk about, are ion channels, which we kind of have already alluded to when we talked about cell membrane structure, but we didn't talk about them in any level of detail. Again, we see that we have a receptor in the protein, a protein receptor in the phospholipid membrane, and this one is what we call a ligand-gated ion channel receptor protein. For some reason, we do not abbreviate this as an LGICR. I don't know why. The other ones get an acronym and these don't. So we just have to say ligand-gated ion channel receptor protein. And they do pretty much what their name says. Okay, so on the extracellular side, there's a receptor. That receptor will bind a ligand. A signaling molecule comes in. these can work in both ways. Okay, so it's a gated, ligand gated. So you think of it as a gate. What are the two options for a gate? Open or close. So some ligand gated ion channels receptors will close the gate, and some ligand gated ion channel receptors will open the gate. This one is showing you an example of one that is closed until the ligand bonds. But it can go the other way where it's open and then closes when the ligand bonds. Does that make sense? So this example is showing you closed, then opens when the ligand bonds. But it can function in the other direction. So in this case, we've got a closed ion channel. We've got some ions out here. The cell needs some ions. So when the ligand bonds, it changes the shape of the receptor. So this channel changes shape so it can be open. And it's going to be open to a specific ion only. So it's not going to open to every ion that's out there in the extracellular fluid. It's specific to one kind of ion. That might be sodium. It might be potassium. It might be calcium. And those ions can enter the cell, and that causes some kind of cellular response when those ions are able to enter the cell. Or stop entering the cell, depending on which kind of channel we're talking about. Then after this, there's not really a picture of this, but if the ligand dissociates, so it unbinds. So when it's no longer connected to that protein, then the channel is going to go back to the way it was. Or it's going to open depending on which kind it was. So an example of this in real life is the nervous system. So the nervous system uses neurotransmitters. So this ligand could be a neurotransmitter. That neurotransmitter is going to bind, it's going to open the channel so that ions can move through. Those ions have a charge, and that charge can cause an electrical signal to pass on to the next cell. So the message is transmitted both electrically and chemically. So the neurotransmitter is a chemical ligand that bonds that will allow ions to come through. Then we have charged ions which build up enough to cause an electrical signal, which will pass through to the next cell, which then can open. Those ions themselves, this is so we talked about ligand gated. There's two kinds of gated ion channels. The second one is a voltage gated. So sometimes that electrical signal will stimulate the cell to release neurotransmitters so that we have the chemical and electrical thing, but sometimes the electrical signal is enough to open the gated channel. So some of the gated channels are voltage gated. So the electrical signals can open and close. So the nervous system is an example. These kinds of channels also exist a lot in the ER membranes. So the membranes of the endoplasmic reticulum also have a lot of ligand gated ion channel receptor to allow things like calcium to go in and out. So those are three kinds of transmembrane protein receptors: GPCR, RTK, and ligand gated ion channel protein receptors. Okay? That's all talking about messages coming from outside the cell and going inside the cell and causing a reaction. You can also, and we don't have a slide for this, so we're going to draw a little line. You can also have messages that are passed within the cell. So instead of intercellular communication, there's also intracellular reception. So the cell doesn't only communicate with the outside world and cells outside itself, it can also communicate with itself within the cell. So this is often that messages are passing to the nucleus. So we're trying to get messages to the nucleus. And sometimes this happens when signaling molecules come through the membrane. So instead of binding with a receptor on the cell membrane itself, the signal molecules come into the cell and they can either communicate with the nucleus or with something that's in the cytoplasm, which might be another organelle. So if it has to come through the membrane, thinking back to cell membrane structure, what kinds of membranes, what kinds of molecules can come through the membrane more easily? So polar molecules. So things that might be able to come through the membrane might be polar. What size are they going to be? They're likely to be small. And some of them might be hydrophobic. Because hydrophobic molecules can also move through the membrane pretty easily. So there are some hormones that fall into these categories. So hormones that are steroid hormones, things like testosterone and estrogen are steroid hormones. So they can pass through the membrane and work with intracellular reception and intracellular communication, and some of the thyroid hormones too fall into this category of being able to pass through the membrane to go into the cell directly without binding to a transmembrane protein. So we talked about three ways that you bind to the cell membrane, but also remembering that things can come into the cell themselves. So that's the first step: signaling, transduction, reception. So binding to this proteins in the cell membrane or coming into the cell directly. What's the next step of the whole process? Is that a hand there? No. After signal reception comes what? We did so well 15 minutes ago. Not yet. What's between, yes, signal transduction. So after we have reception, so we're talking about specific kinds of receptors that are involved in signal reception, we have signal transduction. So we have to be able to pass that message on. And guess what? It's not simple. It's not just like, oh, here's the message, cool, got it. Okay, there's a multi-step process. Lots of places to, I don't want to say mess up, but lots of places that can mess up. So transduction, we're talking on passing on the message. I'm going to try this again. So once the message has gotten into the cell, how do we pass that message along within the cell? So one of these methods is through protein phosphorylation. What do you think that means without talking about it? You can probably guess. What are we going to do? In protein phosphorylation. You're going to sit silently. Phosphorylating means we're gonna add what? Phosphates to what? Proteins, yes. So we have protein phosphorylation. Okay, we're going to add proteins to the phosphorylations. These are relay proteins. So we have a series of proteins and we're going to phosphorylate them and that's going to help send the message on. Some of you are track people. Are any of you relay runners? No relay? Yes. What do you run, parents? 4x4. What leg do you run? Second leg. Okay, so when you're running a relay in track, what message are you passing on? You better get the baton off or you're gonna be in trouble. I've never seen a relay where there's no baton involved.
I've seen plenty of. Say,
I've seen batons be dropped.
I've dropped it.
I have also seen some. There's a reason that's not in the relays. 4x1 is less stressful than the 4x4 is less stressful than the 4x1. But you have to have a number of people and you have to have a baton and you have to pass that baton on. If you drop that baton, then the message doesn't get passed and you're disqualified from the race and you lose. Okay, same idea here. We have a message that we have to pass on. It has to be passed on to every member of the protein phosphorylation cascade, is what it's called, the protein phosphorylation cascade. We have to pass that message on. So if we think of it as a baton, the analogy can only go so far, but if we think of it as a relay with a baton, okay, so we have our message coming in. This is the gun, I guess, that says go. So we have a signaling molecule here. We're using growth factor as an example. I can't even spell molecule. So you're in the starting blocks and the gun goes off. That's the signaling molecule is going to bind. Okay, so it's going to bind to this receptor protein. We'll consider this the first leg of the relay. Okay. Oh. And so we're going to activate this. Maybe we shouldn't consider the relay. We'll still consider that the gun. This is the first leg, is the activated relay molecule. So when this ligand bonds to the receptor, it's going to activate this relay molecule. This activated relay molecule is going to take an inactive protein kinase. I feel like I need a different color. So it's taking an inactive protein kinase and it's going to make it an active protein kinase. I'm going to abbreviate protein kinase PK because it's too long to write out. What do we know that protein kinase molecules do? Enzymes. What do they do? Somebody muttered it, I think. 2 slides ago, 3 slides ago. What does the protein kinase do? Doesn't break down protein. Catalyzes the phosphate, taking a phosphate from ATP and putting on a protein. So it catalyzes protein phosphorylation. So we have an inactive protein kinase molecule. This relay molecule is going to activate it. So now we have an active protein kinase molecule. It's going to take a phosphate group from ATP, and it's going to take this inactive. So this is protein kinase number 1. It's going to take protein kinase number 2. It's going to add a phosphate group to it. So now we have an active protein kinase number 2. This active protein kinase number 2 is going to take this inactive protein. It's going to phosphorylate it, making it an active protein. So this is a protein, not a protein kinase. And that's going to cause a cellular response. So we have the first leg of the relay. Protein kinase is the second leg. This is therin, the second leg of the relay. Protein kinase number 2 is the third leg of the relay, and this active protein is the fourth leg of the relay, and we won. Yay, we got a cellular response. Okay, this protein phosphorylation cascade, so where we're taking protein kinases and activating more protein kinases until we get to this last one, could be much longer than this, but there's only 4 people in a relay, so thankfully there's only 4 people here. But 4 proteins here. It can be longer than that, but that's just, we have a series of relay molecules that are phosphorylated by a kinase. We also have to have the reverse of that. So it's not just that they're phosphorylated. It's not just that they get the baton. They have to pass it on to the next one and be ready to go again. So there are these molecules, which are a different enzyme. And this is where our race relay breaks down, because I don't know of any person in the relay that is involved in this, but these are called protein phosphatases. I did spell it right. And what do you think they do? Kinases add a phosphate group, and phosphatases take it back off again. So they're going to remove that phosphate. That's going to make it inactive. Another protein kinase can come in and phosphorylate it. A protein phosphatase takes it off. So kinases add the phosphate groups. Phosphatases take it off, making it useful again. So it's allowing all the materials to be recycled over and over and over. Those kinases can, phosphatases, those little cycles within the cycle can keep regenerating so that the inactive can become active and again. So we talked about phosphorylating tyrosine as a tyrosine kinase molecule. Most of the proteins in the cytoplasm, most protein kinases will phosphorylate only 2 amino acids. 309, threonine, however you like to say it, or serine. So we activate the proteins, the relay proteins, by adding a phosphate group, and then we inactivate them by taking off the phosphate group. And this is, again, it's called a cascade because one acts on another, which acts on another, which acts on another. And again, this can be longer, they can be shorter. And what ends up happening, the cellular response, cellular response is regulated by the balance or the ratio of protein kinases to protein phosphatases. So if you have more phosphatases, protein phosphatases acting, that's going to take off more phosphate groups, so that's going to inactivate them. If you have more kinases, you're going to activate more. So you can kind of control the rate of response based on which enzymes are more present. And we got to move faster. I'm getting all wound up about phosphorylation. Okay, transduction also happens through the process of secondary messengers or second messengers. These second messengers are small non-protein molecules. So when we talk about protein phosphorylation, we're talking about relay molecules, which are proteins. They're also small non-protein molecules that are water soluble. So they can be molecules, or they can actually be ions also, that act as secondary messengers. So I don't know, this is like handing the baton off to the other team. I don't know. Again, our track analogy only works so far. So we're going to actually talk about this one on the left first. So there are 2 molecules that are the most common secondary messengers. One is called cyclic AMP, and the other one is the calcium ion. And these are part of GPCR pathways or RTK pathways. We're just talking about the transduction part instead of the reception part. So on this side, we're looking at cyclic AMP acting as a secondary messenger in a GPCR. lots and lots of letters. So we've got our GPCR. We've got our ligand. This should be starting to get boring because we've talked about it over and over and over and over and over. Oops. Okay, so the ligand's gonna bond, it's gonna activate the G protein. That activated G protein now has GTP bound, so not GDP anymore. That's gonna bind to an enzyme, and here we're talking about a specific enzyme, which is a adenylyl cyclase. This is an enzyme that's gonna convert ATP to cyclic AMP. So adenylyl cyclase takes ATP and makes it into cyclic AMP. So see, ATP has triphosphate, AMP has monophosphate, so it's going to remove 2 phosphate groups, and it's going to cyclic it, it's going to make it circly. Cyclic, I don't think is a word, but. Okay, then that cyclic AMP is going to be our second messenger. So we've taken the message from a protein, from the signaling molecule, to a protein to a third kind of molecule, a second messenger, which is then going to activate some kind of protein kinase, and it's going to cause some kind of cellular response. So some signaling molecules will activate adenyl cyclase and some will actually inactivate it or inhibit it. So make it not work. So if we inhibit adenyl cyclase, what's going to happen to the cellular response? If this is inhibited, then this isn't going to happen, then this isn't going to happen, then this isn't going to happen. So it's going to slow it down or stop it. On the other side, we see an example of the calcium ion. And another one called IP3 in a signaling pathway, which again is a GPCR. So you've got a GPCR, you've got a ligand bonding. It's going to activate the G protein. That's going to actually interact with this molecule, which is an enzyme, but it's not a transmembrane protein enzyme. It's a peripheral protein enzyme, phospholipase C, which is going to act on PIP2, which is going to break PIP2 into 2 different molecules. One is DAG, which goes on to, this works in other cellular processes, but this IP3 acts as a second messenger. It's going to bind with a IP3 gated calcium channel on the ER membrane. So we've got the ER membrane. There's a high concentration of calcium inside the ER. We need some calcium out here. So the IP3 is going to bind to this gated channel. It's going to open it up so that calcium can come through. That calcium is going to activate other proteins, and we're going to get cellular responses. So that's simple, right? No, it's complicated. You may not love all this detail, but I'm hoping that you're at least a little bit impressed by this, that your cells can do this. Like, this is a complicated communication process. And if something doesn't go right, then something doesn't go right in your body. And your cells are doing this right most of the time. Almost all of the time, they do this right. And it's all of these molecules, like these lipases and the PIP2 and IP3, all of them have to be made by your cells. So they're making these molecules and setting up these very complicated communication methods at a cellular molecular level. And I'm just like, wow, this is amazing. And I can't even think about it. But this calcium, it's hard to comprehend. There's a lot of steps. And so I hope you're at least a little bit impressed by your cells. You should be. I've seen t-shirts that say, like, I may not look like I'm doing anything, but at a cellular level, I'm really quite busy. Okay, this is what your cells are busy doing. Okay, they are making all these components of this and then carrying out these complex reactions to make things happen inside your cell. So different signaling molecules will inhibit or cause these things to happen so the reactions happen. We've kind of alluded to this. When we talk about cellular responses, They can take different forms. We've got 8 minutes. We're gonna go real fast. Okay, a nuclear response. What's inside your nucleus? DNA. DNA has the instructions for what your cells need to do. So a nuclear response can be telling your cell to make mRNA. We're going to talk a lot more about that in chapter 16 or 17. That's just a way for your cell to carry the message out of the nucleus to something else. So this reactions to these signal transductions might be that your nucleus, your DNA starts making mRNA. So this again is showing us this. Here's the signal reception. This is showing us a nuclear reaction. That's funny. It was a nuclear reaction. A reaction in the nucleus, a nuclear reaction. Okay, we've got the reception, the growth factors binding. What is this? Some kind of protein phosphorylation cascade. The last active kinase in the protein phosphorylation cascade is going to enter the nucleus. So it's going through the nuclear pores into the nucleus. And this is what we call a transcription factor, which again, we'll talk more about in a later chapter, but this is inactive. That protein kinase is going to phosphorylate it, making it an active transcription factor. That active transcription factor causes the cellular response of DNA being transcribed into mRNA. That's the cellular response. Is an mRNA transcript, is the cellular response to this growth factor binding to this protein and this protein phosphorylation cascade and this active protein entering the nucleus. That's the response. We can also have cytoplasmic reactions, which just can make cells determine what activity of proteins there is. So are the proteins active or inactive? Are the ion channels on or off? That kind of thing are cytoplasmic responses. So channels open or close, lots of other kinds of responses, but the cell has to be able to regulate all of this. And it can do this in different ways. So one way it can regulate it is the degree of amplification. So when this happens, how many protein cascades are activated? So maybe it only activates one, maybe it can activate 4 or 5. So how much do we amplify the message? How loud does the message get? So it determines the reaction rate by the amount of amplification that happens. Because it has many steps, there's lots of places that you can stop this from happening. If one of the kinases isn't there, or if there's something that inhibits one of the kinases, you can stop this at a lot of different steps. So that's a way to regulate how much is going on. Because you have a lot of steps, you have a lot of places to stop it. You can also use scaffolding proteins, which is what we see down here. This thing is called a scaffolding protein because it can carry different protein kinases. So this scaffolding protein has 3 different kinases on it. When it binds to this receptor, it's going to activate 3 kinases at the same time. So that can cause a more dramatic reaction. And then just by having some way to terminate the reaction. So having some way to stop the whole thing from happening allows the cell to regulate it. It's not just that they have to regulate one pathway, it's that these pathways can also interact with each other. So you have multiple pathways that might be interacting with each other. So binding those signaling molecules can make lots of different things happen. It's also, these are very specific. So signaling molecules can act on different, I don't know where to write this. This is not the best place. Oh, because so signaling molecules can act on different cells differently. So when adrenaline goes out, it's going to act on a liver cell differently than it's going to act on a muscle cell. So even though it's the same signaling molecule, it's going to have a different cellular response. So that also allows the cells, the pathways to have more of an effect. 2 minutes on apoptosis. Okay, apoptosis is programmed cell death. Sometimes your cells have to die. Sometimes this happens. Why? Why do you want cells to die? You want cells to die in development processes. And this is showing you an example of the feet of a mouse. This is a mouse paw. So during development, there's a lot of interdigital tissue, but the mouse wants fingers. This happens in human development too. And so these cells have to die. So these cells in between the digits die off so that you end up with space in between the digits and you get fingers. So in the development process, this is also important for immune cells, the immune system, to be able to kill other cells, to make cells die. And for recycling materials. So the cell wants to reuse materials. It doesn't want to have to make everything from scratch. So being able to recycle materials is important. So if the cell can control how it dies, it can reuse those materials. And we will have to spend a minute on apoptosis on Wednesday. Okay? When is your homework due? When can you register? Do it, if you haven't already. I think so. I think I authorized you, right? You should be good.
I think there's a game on Wednesday, so is it okay if I change that? I won't.
Right, you guys are going. Okay, I knew you were going somewhere. I couldn't remember what. Yes,
Thank you.
and I'll record.
Okay, thank you.
I cannot make this go away. I don't want to see this. I don't want to see more. I want to see this.